In this video we will discuss about, or we will continue our discussion about this high nociceptive neuron relation with M2 type macrophages. In the previous video we have ended our discussion where we checked the spatial descriptions of different immune cells using this codex tumor regions, using this codex spatial system, and here they have shown the quantifications of that, and we can see that for this poor nociceptive or paired nociceptive neuron which are poor and low pain, and nociceptive neuron which are abundant shows a significant difference for this M1 like tumors. So M1 like macrophages and M2 like macrophages, you can see that it is higher when these neurons are abundant, indicating that when the pain is high and the neurons are abundant, then they will attract more M2 like macrophages and also this M1 like macrophages, and the M2 like macrophages will cause the tumor suppressive environment. And so this is, they have validated through the quantifications of the spatial immune distributions using the codex platform. Now they did move to the second studies where they took this HNSCC, that is head and neck squamous cell carcinoma populations. Here what they did is they checked this similarly, similar to that they did it in the previous studies, the pain level of the patients, and then analyze this plasma plus PBMCs and this multiple immunofluorescence. So they analyzed the plasma and PBMCs for data independent proteomics, and then did the analysis whether what are the difference in the gene expressions from the immune levels point of view, and they divide these categories into two different populations based on the stage at which those populations are. So here they divided into this stage 2, 3, and that is not characterized by any stage. And they found out that 12 of them from the stage 2, 3 are belongs to this low abundant, and 13 belongs to the high abundant of the neuron. So when they did the analysis using data independent analysis, they did the data independence proteomics using this site of detector, and they validate using the ELISA and flow cytometry. So from this data independent proteomics, when they check the pathways, they are able to see that the pathways that are upregulated when the neuron are abundant and the pain is also high are this innate immune response, and also this nervous system development in the response to stress, because the cells are in the tumor suppressive environment. So in that case immune cells are not being able to perform its function, that will create a continuous stress on the cells that are within the tumors. You can see that innate immune response seems to be on the lower side, as number of cells or number of signals are lower compared to the other cases. And when they check it for the poor neuron and when the pain is low, they are able to find out that in that case the immune effector response is significantly higher compared to the innate immune response in the high abundant cases. And here the hemostasis and coagulations are also maintained, and the defense response is also there. So the system here, when the neuron is low and pain is low, is working much better compared to the case where neuron is high and the neuron is abundant and the pain is high. And then thereafter we did plot one of the representative image of these PBMCs. They plot Disney plot to shows the different categories of the cells that are present in here, and in the panel they quantified for one of the categories that at stage 2 and 3, that has this well pooled population of this low neuron and pain, and 13 pooled population of this high abundant neuron and high pain. And when they quantified, they are able to see different populations of the immune cells in this. and indicating that in both the cases the immune system works differently because they are at different stages of the cancer. And these things they have further quantified or validated using the flow cytometry analysis, which shows that total CD8 are high in the low abundant cells, indicating cytotoxicity cells are high. But in the case of the high neuron, where more tumor suppressive environment are there, CD8 plus cells are low. This completely makes sense as it will have the immunosuppressive environment, so CD8 cells might have got inhibited, and the other cells are high. That is different kinds of monocyte, classical monocyte, non-classical monocytes, and transitional monocytes are high in the high abundant and in the abundant neuron and high pain area. So this is what they so from this results, this is the rest of the part of the panel A, figure A. So from that they conclude that this both nociceptive neurons might play a role towards this M2 type macrophages, as M2 type macrophages seems to be high when the nociceptive neurons are abundant, and they are low when the nociceptive neurons are low. So this is what they finally conclude, and these things they validated through two different studies. One studies we have discussed in detail in the previous video, and here most of the focus is on the second studies. So that's all for this video. If you need to understand more about their experiments and their statistical calculation strategies, you can go through the reference that has been provided in the description. Thank you for your time.